All right, thanks for the introduction. I'm Brittany Needham, and I'll tell you today about one particular gut bacterial molecule that I've been studying that I'm really excited about, that we've been able to take from observations in a clinical and mouse context, and then move it into a very controlled environment in mice to study, and then come full circle and move it back into the clinic and see some exciting preliminary results there. So, of course, as all of you know, but this is a little out of context from the previous talks, the gut microbiota is the collection and community of microbes in our gut, in our intestines. That comprises a lot of bacteria, viruses, fungi, archaea. I will talk about bacteria today, and I often make the mistake of saying microbiota, but I'm really only talking about bacteria. And any one of us have up to 1,000 bacterial species living in our guts at any given time. It's a very dense, complex, vibrant ecosystem, and there's a lot of signaling that occurs between microbes in our gut and between the microbes and our tissue. If you look at these electron microscopy images up at the top, you can see some bacterial cells really get very close right along our epithelial cells in our gut. And it's easy to imagine that there's a lot of bidirectional communication that's occurring along this interface. And a lot of the things that we do, or factors that are in us and about us, affect our microbiota: what we eat, how much we exercise, our lifestyle, our age, and genetic background, things that we can't control. And as we are altering our microbiota and the composition of microbes that are present in us, in turn, those microbes are affecting us. And we know that they're very important. Lots of benefits come from having a gut microbiota. I actually gave a talk yesterday, and someone asked me, what do we do to keep our guts clean from any bacteria? And that was after my talk, so I thought, I really failed at driving home the point that we all have these microbes and that they're very important and playing a lot of roles in our nutrition. Bacteria break down complex plant molecules that our bodies can't, giving us nutrients and vitamins, and protecting us from pathogens that may invade, because this ecosystem is dense and already taking up all these niches that a pathogen could take. It's also crucial for the proper development of our immune system and our nervous systems. So we know that the gut microbiota is playing a role. We know that it's very important in many host systems. But moving from correlations of knowing this and having observations of changes in the microbial populations between different patient groups or in different conditions has been very difficult. To move from this correlation of changes to actually showing a causation that these changes in the microbiota matter and they can be causative, more than just a coincidence that they are occurring. And one thing that makes this even more complicated is the fact that this dense, complex population of microbes has a lot of redundancy. So we might have two microbial populations, which here I've colored them 2 different colors, but that's just to represent that maybe the sequencing, or if you made a list of every bacteria that was in your gut, might be very different from another person's list of bacteria in their gut. But that same benefit to you might be the same. So multiple microbiotas and multiple profiles can fill these same functional roles in our guts. And so how do we tease out that 10 bacteria are all able to perform the same roles, and how do we know which ones matter? And so I've approached that. In my lab, we're really interested on looking at the signals that come from the microbiota as a whole. And as we look at the microbiota as the source of a vast pool of signaling molecules, we can identify signals that are important and then maybe trace back to the microbiota and a particular bacterial species, if there may be one, or to the brain. And I have this slide, which is more complex than needing to look at, but just to remind us that in the microbiota there are over 22 million unique genes and many enzymes capable of modifying a diverse group of chemical structures. And then we have all these microbes in our gut that are constantly coming into contact with our own host molecules, with molecules from various bacteria and microbes in the gut, and everything we eat passes through this filter of microbes and can be chemically transformed into these other small molecules and signaling molecules that then get into our system and can be distributed throughout our body and reach in our brain. These signals can be harmful, they can be beneficial. But I really view them as one of the groups of major players of signaling between this gut-brain axis that I'm interested in. And I also think it's a very, encouraging approach to take, which drives me in my lab, because these microbes are in us, they're of us, they're part of us and influencing us, but they are separate from host enzymes and host systems. And so drug targets that can limit a bacterial substrate or an enzyme that modifies a host or dietary product and causes these signals, or the signals themselves, all three of these kind of substrate, transformative enzyme, or microbial output can be targeted, ideally with a lot fewer side effects to the host and maybe a very tractable therapeutic option. So in the project I will describe today, we set out to ask, are there signals specifically from the microbiota, and are they influencing the brain, and if so, how? And this is, of course, in the context of ASD, but this work quickly became focused in on the anxiety aspect as a comorbidity of ASD. And the first thing I was able to do was, we had a large cohort of samples, and we had Metabolon do global untargeted metabolomics. So asking the question, what small molecules and chemicals are present in these samples from ASD children compared to some neurotypical controls that we had. And we were able to do plasma samples and paired fecal samples from many of the subjects. And we were able to get a really large data set, which was an advantage over some previous studies that, if you target a few molecules, you can't look at the whole picture. So we were able to get a really quite global picture. And surprisingly and excitingly to me, with my interest, there were several bacterial molecules that really stood out in this data set. In fact, 4-ethylphenyl sulfate was the highest fold change molecule in between our two groups of samples. And when we did a machine learning analysis to ask kind of which molecules are differentiating these two patient groups or two sample groups, 4-EPS or 4-ethylphenyl sulfate was in the top 25 of these molecules. So we see a bacterial molecule come out and be highlighted in this global metabolomics study where we tried to look at every molecule that was present. And this was exciting also because this same molecule was observed to be elevated in a mouse model that actually Stephen helped set up in his lab at Paul Patterson. We just realized that this morning. And in some other mouse models of atypical neurodevelopment that I've worked with. So we were emboldened to continue this molecule and see if it has an effect. So we engineered and identified the biosynthetic pathway of this molecule. So that's 4-EPS at the end there. And the host cannot make this molecule, so it's not present at all in a mouse that doesn't have a microbiota, and it's a tyrosine metabolite. We've identified other pathways now. Microbes, with their diversity and creativity, can use other molecules to end in this product. But we can take tyrosine and convert it to 4-ethylphenyl by the bacteria. And then once it enters the host, it's quickly sulfated by a variety of sulfate transferases, actually, into 4-EPS. And so we identified and confirmed this bacterial pathway, and we engineered it into some bacteria. So we have two bacterial sets. One that I'll call 4-EP minus, which is just bacteria that don't produce this pathway. No 4-EP is produced by these bacteria in any circumstances that we've identified, and no 4-EPS results in any host system. And then 4-EP plus, which is just bacteria that are overproducing this pathway, and so otherwise they're isogenic, and so they're the same bacteria, and gives me a really good opportunity to carefully control and reduce all the complexity that I told you about in the microbiota, but still have it in a host system. And so we took germ-free mice, which are mice that have absolutely no microbes in or on them in any way. They live in these bubbles, and colonized them either with the non-producing bacteria, 4-EP minus, or the producing bacteria, 4-EP plus. So these mice are then exposed to the molecule. There's no other difference. These are wild-type mice, and they're totally the same, except for they have bacteria that are either producing or not producing this one molecule in their intestines. And then we let the mice hang out for a while. Once they're adults, behavior test them and do other biochemical and other analyses. And so just to establish this model, we confirmed that indeed mice in the 4-EP plus group have 40P in their feces, and in their serum, urine, and in their brain, we can detect this 40PS molecule that I was interested in studying. Okay, so the first thing we did, well, that's not the first thing. First thing, I looked all over these mice, and they seemed to be pretty normal. Then we looked at the brain and did some functional ultrasound imaging, which is similar to functional MRI, which I think probably everybody in the room is familiar with. In functional ultrasound imaging, it's really an ultrasound probe. It's extracranial, and the mice are anesthetized but alive, and over a period of time, you measure basically the blood flow in the brain and can correlate brain activity across regions. So you overlay the map and make a correlational matrix, similar to what we saw in Naveen's presentation. So these blue and colorful squares here are just correlational matrices with brain regions along the left and on the bottom. That is too small for me to list them all. And just by eye, I think you can see that across the top row, the 40P minus mice have more blue, less functional connectivity, less correlation between these different brain regions than in the 40P plus mice. So we're observing higher functional connectivity, and the regions that these are attributed to are summarized along the bottom here. So the first take home was just it's exciting that this one little molecule made in the gut of these mice is sufficient to affect the brain in some way. And then when we looked closer at some of these brain regions, we observed that the amygdala and the hypothalamus and some of these limbic regions of the brain that play many roles, but one of which is involved in anxiety and fear responses, are altered in how they're responding in this setting. And so to move towards a higher resolution experiment, we did another brain imaging study where you inject a radio-labeled tracer, and it gets trapped into metabolically active cells, and it's a way to distinguish brain activity. And so in these images here, this is a summary of all the mice. So anywhere that you see red or yellow, these warmer colors, means that there was more elevated brain activity in those regions in the mice exposed to the molecule over the mice that had no molecule. And so, for example, like the hypothalamus and the BNST and the PBT all showed elevated brain activity regions of the amygdala when they had been exposed to this bacterial metabolite in their gut. We use this experiment to inform us, what brain regions should we be looking at to try to figure out how this molecule that's produced very far away from the brain is then affecting it. It was a black box kind of between those two regions of the mouse. And when we studied these mice a little bit further, we did a battery of behavior tests, doing motor testing, social behavior, repetitive behavior, all of the things. We did all the things. And the test that we saw the strongest phenotype in was anxiety response. So this is the elevated plus maze and open field test, which we've heard already today. They take advantage of the fact that mice tend to explore. They're curious, but they're also prey, and they're nervous to spend time in these exposed, open, brightly lit regions of the apparatus. And we see that 40P plus mice show more of an anxiety-like behavior in these tests. And then also the marble bearing test, which mice are given a grid of marbles on their bed, and mice that have higher levels of anxiety-like behavior dig more and bury these marbles more. So these are a few ways to measure this behavior. And consistent with the brain imaging studies that we did, we saw changes to the brain regions that may control these behaviors, and we do see changes to the behavior. And so we did some RNA-seq on the brain regions that we see changes in. And the thing that, it was surprising, we didn't expect this at all, but the thing that we saw the highest change in was in profiles of oligodendrocytes. And specifically that immature, young precursor cells to oligodendrocytes were elevated, and mature myelinating oligodendrocytes were decreased. And I say the cells, these are actually just their genetic profiles of these cells, genes that are indicative of these cell types. And just to show you a few examples, so like MOG is a very famous one, is decreased in our mice that are exposed to this bacterial metabolite. And if you don't know what oligodendrocytes are, and I didn't when I first got this result, so we came at this quite by accident, they're very cool cells. They're the myelinating cells of the brain, so they are insulating all the axons and producing the white matter in the brain. And these are some in vitro images of just pure culture. But so a precursor cell is a very different morphology than a mature cell that then is producing this myelin and excreting a lot of this lipidated membrane that then can wrap around an axon when it is present. And there's some indication in the literature that microbes in the gut can influence this in the brain. So a germ-free mouse that has no microbial influence whatsoever, in their prefrontal cortex have a lot more myelination. And this is in these images here, this is like an axon coming out from the plane of the screen, and then it's a cross-section. And so that dark gray circle, I don't know why I'm pointing with my hand, this dark gray circle here around the axon is the myelin. And you can see by eye that germ-free is much thicker than the conventional mice that have a normal level of bacteria in their gut. And the same thing can happen when you treat with broad-spectrum antibiotics for a period of time and the mice no longer have any microbes. Then there's similar shifts in these oligodendrocytes and in their myelination. And another thing that's pretty exciting about these cells is that, at least for me in my neuroscience classes, I learned that myelination occurs kind of in development and it's very important, but then it's largely static and maintenance mode. But a lot of new studies are really showing how important myelination in oligodendrocytes may be in a lot of different behavioral cases and a lot of different disease states. And throughout adulthood, as we continue learning, and even in old age, we have a lot of myelin remodeling that's very important. And so these studies kind of are indicative that maybe that is playing a role and we should follow up on this. And so we looked at the cells themselves rather than just the genetic profiles, and we observed that by staining in the presence of this metabolite, 4-EP, we see elevated levels of a marker of early oligodendrocytes and decreased levels of a marker of mature oligodendrocytes, and that's quantified here just as a ratio for simplicity. And we saw that in many methods, like flow cytometry. We did Western blots for myelination, and we observed this quite consistently across these mice. But even further to go functionally, is this actually affecting the myelination? And so we did some EM on a region of the brain where the axons are quite in a highway in parallel, and then take a cross-section. And so I think it's maybe the most obvious in these top two images here. So 4-EP on the left, 4-EP minus on the left, you can see all these dark gray circles is the myelination around every axon in this region. And then 4-EP plus over here actually has the same number of axons in this image, but the dark gray circles are quite a bit more sparse and there aren't as many. And so you really can observe in this image and here quantified that we have a higher percentage of unmyelinated axons in the presence of this metabolite. And that's also observable in how thick the myelin is, like longitudinally. This is along an axon. And I know I'm just showing you one image. We did take a lot of images and count quite a few cells across multiple mice. But in these 4-EP minus has a lot thicker with more myelin sheaths wrapped around these axons. And to go one step further to really convince ourselves that myelination is affected in these mice, we did some MRI imaging of the whole brain and observed through diffusion tensor imaging that the myelination is less organized in the mice with 4-EP produced by the gut bacteria. And then we were able to treat with a drug that was designed, these experiments were designed to link the phenotypes between this bacterial metabolite that's produced and affects behavior and affects the oligodendrocytes, but are the behavior and the oligodendrocyte phenotypes linked to each other? And so when we induce through a drug oligodendrocyte differentiation, and so they're restoring the oligodendrocytes to control levels, we see a restoration in the behavior that this drug that induces oligodendrocyte differentiation and myelination is then restoring the behavior to normal levels. And so I've kind of been summarizing as I go along, but it was exciting to take this molecule and then be able to see in a controlled way that it is having an effect on the brains and behavior of these mice. And I'll tell you one more quick aspect of this story is that how I said that is a very attractive therapeutic target in my mind. We know that these microbes are producing these metabolites, and then they get into the host, they get circulated around, they can reach the brain. what if I can add a drug that will then adhere to the metabolite and be excreted before it could ever get into the host system? And there are many ways to approach altering the levels of this metabolite. This is what I chose to do first. And this drug doesn't get into systemic circulation at all. It just passes right through. In mice, it's actually visually very obvious because mice are so small. So we use these same groups of mice, 4AP minus and 4AP plus. 4AP plus is orange, and then when we give them this drug, we see that the drug does decrease the level of the metabolite. And in mice, it restored behavior largely to the control mice. So this orange is the 4AP plus mice, and then on the drug, the blue, we see an elevation of willingness to explore these vulnerable regions of the apparatus, of the mazes, and a less of a tendency to bury a lot of marbles. And we partnered with Axial Therapeutics, which there are actually notebooks around the tables for this company, and did a clinical trial where teenagers with autism in Australia and New Zealand were placed on this same drug for 8 weeks. And over the course of treatment, we did metabolomics and behavior testing. And at baseline levels in orange, we see a baseline and elevated levels of 4-APS. And then at the end of treatment in blue on the right, we see a decrease in this metabolite. So in these teenagers, this bacterial metabolite was decreased by the drug. And I just couldn't even believe it, honestly. They had lower levels of anxiety scores and irritability. So this is, again, the baseline in orange, and then the end of treatment in blue. And then we had a washout period where for a month they weren't on the drug at all, and we see some bounce back there. And I should say this was a phase 1 open label trial, only on 29 individuals, and there's a lot more to be done and a lot further to go, but it's very exciting for me to see that in humans this metabolite might actually be playing a role, and it's not just specific to some strange thing in mice that at one point started out as a germ-free mouse, which is a very abnormal setting. And so we're able to see this oral adsorbent is helpful in the clinic as we reduce levels of this harmful bacterial metabolite in the system. And our future work is to answer all of the questions that remain that are in your minds now, like, is this acting directly in the brain? What metabolite, like, is it the 4AP or the 4APS or some other breakdown that's actually playing the role? And what is the molecular target of 4APS? And those are all things that we're focusing on now. We have some exciting preliminary data that needs a lot more work. And of course, like everyone else, this work was a group effort, and I have everyone listed here that played a role. And thank you for your time.